Hello Imbibers, welcome to today's session. A very good afternoon. So children, today we are going to discuss ahead of the biological classification. In the yesterday's session we have discussed about that how the classification came into the limelight, how different scientists put forward their own classification system. It was 2 kingdom classification, 3 kingdom, 4 kingdom, 5 kingdom, and yet there was also 3 kingdom classification. So we have discussed all the kingdoms of classification, and we have also discussed the basic drawbacks that every classification possess. So now we know that none of the classification is perfect, but yet we are still having a whole share of 5 kingdom classification. We still believe that's the most explained, I would say that is the most explained classification, which is the 5 kingdom classification. Okay, and it is only elaborated in all the books, in all the sections. So today, according to the 5 kingdom classification, we are going to start the first kingdom of classification, which is the kingdom Monera. We are going to see how the basic characteristics of the kingdom Monera exist and how it is being classified, what characteristics it possess. Okay, so now we know Monerans, typically bacteria, all the characteristics will revolve around the bacteria only, and we are going to discuss about why they were kept separately from the Protista also. Okay, so let's start our today's session. So before we start, let me introduce you to myself. I am Anam Khan. I'll be your botany expert for the session 2023 and 2024, and we are going to discuss all the amazing and interesting sessions related to the plant. Okay, so let's start our today's session. Talking about the kingdom Monera, children, so the very first thing going to discuss all through these sessions is that we're going to discuss about kingdom Monera. We're going to discuss everything about related to kingdom Monera. Then we're going to discuss about Protista, fungi, plant kingdom, of course, and the kingdom Animalia will be discussed about your zoology ma'am, and the virus, viroids, plants and lichens. All these sessions will be taking place regularly on our Embibe app, okay, in the proper sequence. And the concepts today would be covered. We're going to cover all the general characteristics, the shape of the bacteria and the overview of the bacteria. We're going to discuss all the things today. Okay, so let's start our today's class talking about the Monerans, children. What are exactly kingdom Monera consist of? What are the bacteria? So children, as the life on earth revolved around a single million years ago. Simply, when the life was existing on the earth, it was said the very first organism to be existed on our planet was single celled organisms. Okay, of course, the unicellular organisms. In the unicellular organisms, the digestion, excretion, respiration occurs within the cell. Why they were so efficient? Why we say that their whole existence is related to the single cell organisms? Because every particular process which is required to sustain a life or to sustain life on this planet was done in one single cell. Very low maintenance. Digestion, excretion, respiration, all the processes was done within the one single cell. I would say not a surprise that if one single cell is able to do all the particular necessary processes, it would be very easy for the existence. So yes, the Monerans were existing on the planet only because the whole one unicellular organism was capable of doing all the processes. Okay, and yes, they cannot be seen with our naked eyes, hence they are called microorganisms. Yes, they are completely microorganisms because you cannot see them with your naked eyes. I cannot see. We have to use certain equipments. We need microscopes to see these organisms because they are not visible with the naked eyes. So how very tiny, single cell, absolutely tiny, we cannot see them with our naked eyes. Simple, very simple to basically accept it. Okay, and the unicellular existing, which are the bacteria, protozoa, algae, fungi are some of the unicellular organisms. Okay, fungi, of course, the yeast is the only unicellular organism. The protozoa, algae, bacteria are some of the unicellular organism existing on our earth, which basically gave the whole idea of having all the processes in one single cell. So this is just a bit of an introduction that how this unicellular organisms were capable of doing all the things and able to exist life, to have life like millions and millions of years ago. And they were also we have also said that these were the first organisms to exist on our planet. Before that it was nothing. These were the first organisms to exist, to have a life on this planet. Okay, moving forward children, talking about our especially the kingdom Monera, the features of the Monerans. What are the basic feature or what are the basic important features which are unique, which led the scientist or Whittaker or any other scientist to put them all them into one single kingdom and one single category, which is the kingdom Monera. Talking about it, that these are unicellular organisms made up of one single cell. They are made up of one single cell. They have 70S ribosomes. Talking about the ribosomes, which are majorly used for protein synthesis. The ribosome is used for protein synthesis is 70S. Okay, it is used as 70S subunit. Siderberg unit is called 70S. You will have this detailed explanation of this ribosome in your cell structure and function chapter. Okay, right now for you to remember only this thing, that these Monerans have 70S ribosomes. Okay, the DNA which is present inside it, having DNA or the nucleus having the DNA does not have a membranous structure covering the nucleus. It means the nucleus is completely lacking the nuclear membrane. Okay, next the mitochondria, the lysosomes, the plastids, the Golgi apparatus, the endoplasmic reticulum is completely absent. How can you even expect only to have such organelles in one cell? Okay, because the more complexity increases, more difficult is for any organism to exist or to survive in the extreme condition or to adapt in certain condition. So keeping it very simple organization, they do not have any of these organelles. Okay, they lack completely mitochondria, they lack Golgi apparatus, they lack lysosomes, ER absolutely absent, and of course the nucleus even doesn't have a nuclear membrane. Okay, the reproduction. Okay, the reproduction, how they do reproduce? Simply they reproduce, of course they have to do it asexually by the binary fission or budding. What is binary fission? It means the whole organism, the nucleus will divide, then finally the whole body divides into two. Okay, then again those two organisms will divide. Okay, the complete division takes within one cell, this completely asexual, the other partner, the other I would say the other partner is not involved, the opposite sex is not involved, no fertilization, nothing. Simply very simple, absolutely simple method of asexual reproduction, which is the binary fission and budding. Okay, these are environmental decomposers. Decomposers means they completely destroy or degrade the dead and decaying matter. They completely degrade the dead and decaying matter. Okay, they completely do that. So that is why they are also decomposers. Mode of nutrition, how they obtain their food? They are completely autotrophic because they are pigmented. They can be parasitic, maybe existing as a host on any other living organisms, can be heterotrophic, of course obtaining food from the other organism, and can be saprophytic, existing on the dead. So mode of nutrition is several. They can be autotrophic, they can be heterotrophic, they can be saprophytic, they can be parasitic. They can obtain food from any method of their nutrition. They obtain their food from any method they wanted to obtain food. So that is why when you have so much of variety or for obtaining food, or for basically having different, you can adapt into certain condition, that you can have your food, have your nutrition in any condition, that is make you more flexible to adapt in a certain condition even when that condition are completely harsh and adverse. So these are the few characteristics of monerans which we have discussed. The basic characteristics they possess are these. Okay. Next, talking about more about the Monera and monerans, we again going to talk about the general characteristics which are more precise. It means the prokaryotic in nature. Yes, prokaryote means primitive nucleus. Prokaryotes means primitive nucleus. Membrane bound cell organelles are completely absent. Just now we have discussed the ER, the mitochondria, the Golgi bodies, the lysosomes, all the membrane bound organelles which possess membrane are completely absent, and the non-membranous organelle will be present, and the ribosome is of 70S. Cyclosis is completely absent, and single enveloped system is there. Single enveloped system would be there. Double envelope means there will be double membranous structure, outer and inner membrane. But when we talk about single envelope system, it means only one membrane is present. Okay, one membrane is going to present all over the particular body, not a double membrane. Inner and outer membrane is a double enveloped. Single enveloped means only one covering, one layer is present. These are the more additional features which the monerans possess. Okay. Moving forward, children, talking about the question we have what type of ribosomes are present in Monera. Just now we have told you what kind of ribosomes are present in kingdom Monera. Come on, quickly tell me what type of ribosomes are present in Monera. Yes, 50S, 70S, 60S, 80S. Yes, children, just now we have told you. Come on, quickly answer. The correct answer would be the 70S type of ribosome is present in the monerans, in the bacteria or unicellular organisms or prokaryotic organism possess 70S type of ribosome for the protein synthesis. Okay. Moving forward, children, talking about more about this. Yes. Now comes to the most important thing going to discuss about the monerans is how their cell structure is formed. What is the cell made up of? The cell wall is made up of peptidoglycan except in mycoplasma. Please remember always the exception. So, the cell wall of monerans is made up of peptidoglycan, and basically this peptidoglycan, this cell wall is absent in the mycoplasma. Okay, they show both autotrophic and heterotropic mode of nutrition. Just now we have only discussed about it, that the monerans show both kind of nutrition processes, which is heterotropic as well as the autotrophic. The primary method of reproduction is asexual reproduction. They show unique way of sexual reproduction through parasexuality. What is a parasexuality? We will discuss about it, but it is a method, it is a false, I would say false sexual reproduction. It is a false sexual reproduction, and they sometimes do show the parasexuality, okay, which is a false sexual reproduction, and they are completely cosmopolitan in nature, having simple cellular structure and complex behavior. It means cosmopolitan means they are present everywhere. You cannot see them with your naked eyes, but yet they are present everywhere. The microorganism, bacteria would be present in this. Okay, not a problem. Your phone might having a lot of bacteria, but you cannot see it from your naked eyes. Okay, that's why it's absolutely important for you to always wash your hands before and after eating, or whether you do any work, you need to wash your hands regularly to avoid the accumulation of bacteria, so it does not cause any skin problems or any microbes does not get into your body. So the reason these are cosmopolitan, they are present everywhere because they are completely microscopic, simple organization and complex behavior. The cell structure is absolutely simple because they are made up of only single cell, but they do show the complex behavior. Okay. So these are the few things. Talking about the cell wall, let's elaborate the cell wall more. Talking about the cell wall, children, very important. When we talk about bacteria, Monera cell wall is absolutely important to know that what exactly this is whole consists of. The whole bacterial cell wall is made up of peptidoglycan. It is made up of peptidoglycan. Okay. Peptidoglycan, what it is? Peptidoglycan or murin. Children, please remember these names. These names, the peptidoglycan or murin is a polysaccharide consisting of amino acids that forms a mesh-like peptidoglycan layer outside the plasma membrane, most bacteria forming the cell wall. Very interesting. What exactly, how the cell wall formation is taking place in the bacteria? Talking about the cell wall formation, the peptidoglycan or murin, which is a polysaccharide, okay, polysaccharide, okay, chain of carbohydrates is called polysaccharides. Polysaccharide consists of amino acids that form a mesh like peptidoglycan. Peptides plus glycogen. Peptides means proteins plus peptidoglycans means proteins plus glycan means glucose. Okay, which is peptidoglycan. So chain of amino acids and chain of glucose together creating a mesh like structure around the plasma membrane, creating a cell wall. Clear, children? So the whole peptidoglycan consists of both the amino acid and the carbohydrate. The peptidoglycan is a combination of proteins and carbohydrate. So that is why a polysaccharide, a chain of amino acid and a chain of carbohydrate together create a structure around the plasma membrane, and that is what we called is as a cell wall. Okay, I hope this is clear that what exactly creates a cell wall near the plasma membrane, the peptidoglycan, or we can also call it as murin. We can also call it as murin plus or peptidoglycan. Let's see what are the components existing in the peptidoglycan. Talking about the components which are existing in the peptidoglycan, we have peptides, amino acid, L-alanine, D-alanine, D-glutamic acid and L-lysine. These are all amino acids because the peptidoglycan consists of both proteins as well as the carbohydrate, and proteins are nothing but a long chain of amino acid. Peptides plus glycan means proteins plus carbohydrate. Okay, carbohydrate. So peptidoglycans means, so these are the amino acids present in the cell wall or bacteria or the monerans. Then the talking about the carbohydrate, it is a modified sugar. It means N-acetyl glucosamine and N-acetyl murmuric acid. So these are the two components of the peptidoglycan which makes up your cell wall, which makes a whole covering around the plasma membrane. I hope this is very clear. I don't know what exactly would be more explanatory to tell you that the cell wall is consisting of peptidoglycan. These are the components present in the cell wall which is creating a mesh-like structure around the plasma membrane. Clear? Is this clear, children? Any question? Anything you want me to repeat only? I will repeat, not a problem. Come on, quickly answer. Anything you want me to repeat? Any part which is not clear? Okay, So this is all about the cell wall. This is all about the structure of the cell wall. This is about how the cell wall is constructed of, because this is absolutely important. And now if you remember, whenever we talk about the bacteria, we talk about the staining also. The bacteria are also further divided on the basis of the stain, on the basis when they can take off the stain or they don't take the stain if kept in certain staining chemicals. So the bacteria on the basis of their stain, on the basis of their cell structure, or I would say cell wall structure, are further divided into two parts, which is the gram positive bacteria and the gram negative bacteria. Okay. So children, the gram positive bacteria and the gram negative bacteria have two different types of basically the staining on the basis of the stain. They are particularly divided into two parts because they have whole composition in their cell wall is absolutely different. Talking about the gram positive bacteria, the tetrapeptide chain joins the pentapeptide chain. Okay, while talking about the gram negative bacteria, tetrapeptide chain joins with other tetrapeptide chain. Imagine tetrapeptide chain joining with pentapeptide chain, but here tetrapeptide chain is joining with the tetrapeptide chain. Okay, next there is an indirect linkage with each other. Two chains are completely indirectly linked with each other. When we talk about the gram negative bacteria, they are completely having a direct linkage. Okay, as you can see children, the thickness of the cell wall is from 150 to 200 angstrom, 150 to 200 angstrom, and the thickness of the gram negative bacteria is 70 to 120 angstrom. Okay. Next, the lipid content in the cell wall is quite low, while the lipid content in the cell wall is 20 to 30%. Okay, there is also a lipid content in the cell wall which is completely very low in the gram positive bacteria, while in the gram negative bacteria the lipid content is little bit high. So this, on the basis of their staining mechanism, they have also composition related to the whole cell wall creation. These are the also basis of their how basically this cell wall are completely different from each other. Okay. So this is gram positive bacteria, the other is the gram negative bacteria. Okay. So see how the cell wall has further created a whole division inside the monerans only. The whole bacteria family is completely divided into two more aspects, one which is the gram negative, the other is the gram positive, on the basis of their cell wall structure. So children, cell wall is absolutely important characteristics when it comes to the bacteria. Okay. Since they do not have a very complex structure, so whatever they have is having a very complex organization. They might not have a complex structure, but they yet have a complex organization, because the ability to have all the processes in one cell is very unique. So according to that, they have a very difficult, or I would say not difficult, a very different cell organization. Clear? So this is about the gram negative and gram positive. Talking about the question, let's see. The peptidoglycan is a characteristic constituent of the cell wall in which component of cell wall is a peptidoglycan? What do you think? Come on, quickly answer. Come on, quickly answer. Yes, the peptidoglycan is a characteristic constituent of bacteria and cyanobacteria. Yes, majorly, because in the Monera everything comes up protista, not possible, not archaebacteria and eubacteria, not eubacteria or unicellular eukaryotes, prokaryotes. The correct answer is bacteria and cyanobacteria. Okay. The cell wall is the outermost covering of the plant cell, bacterial cell, fungal cell. It is present outside the plasma membrane. Next, what are the types of bacteria existing? So, based on their shape, the typical bacteria is having, based on their shape, based on their staining and types of respiration. So, types of bacteria which are existing is on the basis of their shape, on the basis of their staining and on the basis of their respiration. Okay. So, as you can see, different shapes and sizes. We have studied about the gram negative, gram positive, anaerobic and aerobic. You know, when we talk about the aerobic, it means the respiration which takes place in the presence of oxygen. When we talk about the anaerobic, the respiration takes place in the absence of oxygen. Now, talking about the shapes of a bacteria, let's see what the different shapes of bacteria existing. Okay, so these are the typical shapes in which the bacteria can exist. Children, listen carefully. The cocci, the bacilli, the vibrio, the spirillum, the filamentous, the stalked, the budded. So, these are the few shapes in which a particular bacteria can exist. It could be cocci, it could be bacilli, it could be vibrio, spirillum, filament, stalked, budded, etc. Okay, talking about the cocci, bacteria are spherical and oval in shape. The bacteria are typically spherical or oval in shape. Okay, talking about the bacilli, these are rod shaped with or without flagella. These are rod shaped with and without flagella. Next are the vibrios. These are kidney shaped small bacteria which flagellated at one end. Don't worry, I will show you the exact picture how they look like. Don't worry, the next slide consists all the kinds of bacteria. Do not worry. First, you need not to know that vibrios, these are comma shaped or kidney shaped bacteria. Next is the spirillum. These are spire or coiled shaped bacteria. Next is the filament. The body consists of small filament like fungal mycelia. Next, they are stalked. The bacterium possesses a stalk. Then they are budded. The body of the bacterium is swollen at places. So, these are the few shapes of bacteria existing. Let's see. I'll come to this slide again, but I can let you show this. This is I wanted to show. This is bacilli. This is how it looks like. Can you see? This is a chain of bacilli. This is a bacilli with the flagellated rods. This is a spore former. Can you see? These are the cocci. These are diplo, tetrad, staphylo, sarcinia. These are spiral shaped, the spirilla with the flagellate and others. So, these are the different shapes existing in the whole bacteria. As you can say, these are the bacilli, cocci, vibrios. Okay, these are the vibrios, as you can say, kidney shaped. These are the spiral shaped. Okay, spirocheates. These are the different kind of a structure existing in a bacteria. They can exist in any kind of a form. Single also, in this aggregated form also they can form chains, also can be tetrad structure also, or they can be double tetrad or sarcinia also. Okay, flagellated, non-flagellated, they are completely their whole existence they wanted how to be. Okay, flagella is definitely for the movement only. So, these are the different shapes and sizes of bacteria existing in the whole Monera plant, Monera, kingdom Monera. So, children, these are the whole shapes of bacteria present. So, what we have already discussed, we have discussed about the cell structure, we have discussed how simple cellular organization, simple complex organization they have a simple cell, yet they have a complex organization. We have seen about the gram negative and gram positive. We have seen what is the cell wall consisting of, how it is constructed, what component is consisting of. Next, we have seen the different shapes of bacteria existing. Okay, these are the few characteristics which were taken into consideration, and these are the few reasons that the bacteria, the Monera, have given a unique kingdom called the kingdom Monera. Yet might be they are primitive, but they show a very advanced kind of an organization because they are able to have all the cellular processes within one cell. Okay, moving on, children. Next is a question which is, cocci are Which kind of a bacteria? Cocci are rod shaped, comma shaped, long twisted, spherical. How? What? How they look like? How the cocci are looking like? The cocci, how they look like? Rod shaped, comma shaped, long twisted, spherical shape. How they look like? Very simple. Just now we have discussed it only. Lakshita, Tanmay, Govind. What are the cocci shaped? Simple children, when we talk about the cocci shape, it is completely spherical shaped bacteria. Spherical shaped or oval shaped bacteria are cocci. Okay, cocci are called the spherical shaped bacteria. The next question is, the DNA replication in bacteria occurs. I know this is not something we have discussed, but you will discuss in the reproduction part. But please try to at least answer. Just give a bit of a common sense that what do you think? The DNA replication in bacteria, it occurs within nucleolus prior to fission, before the division takes place, just after, before the transcription, or during S phase. You know the bacteria divides like through the binary fission or the budding. Though before division, do you think the replication will take place, or after the division the replication will take place? Very simple. I made it simple for you. That what do you think the replication will take place? Yes, of course, children. Whenever there is a replication, it has to be done before the cell undergoes the division. So, before the fission takes place, before the bacteria divides into two, the DNA replication should take place. Very simple, a very simple answer. I know. I will clearly give you the hint also. The next question, in the Whitaker's 5 kingdom classification, all the prokaryotic organisms are grouped under which kingdom? According to Whitaker classification system, the prokaryotic organisms are grouped under which? The prokaryotic organisms are grouped under which? Very simple. Come on, quickly answer. Quickly. According to 5 kingdom classifications, the prokaryotic organisms are grouped under kingdom Monera. Just now we have discussed about it. The kingdom Monera is basically the kingdom which consists of all the prokaryotic organism, primitive kind of an organisms. The next question which comes is chemosynthetic autotrophs energy to prepare food is obtained from? Chemosynthetic means which synthesize their own food with the help of chemicals. So, what do you think the chemosynthetic bacteria obtain their energy from? Chemosynthesis, not photosynthesis. Please bifurcate the term. Chemo means using of certain substances. Almost giving you an answer, but please try to attempt it. This is very easy. What do you think? How this chemosynthetic autotrophs and obtain their energy from? So, children, simply the chemosynthetic, they obtain the energy from the chemicals. Chemical energy is responsible for the chemosynthetic bacteria. Moving forward, next question is, Monera comprises of prokaryotic organisms like bacteria, which is the oldest form of the earth. Do you think the Monerans are the oldest form? Do you think the bacteria are the most primitive organisms to have the life on the earth in the very first place? Do you think that? True or false? Absolutely, absolutely true. Yes, yes, because a very single structure was able to perform all the processes, and that is the reason we considered that the bacteria was able to survive in an extreme condition, and it was present on the earth millions and millions of years ago. Okay, so children, we have already discussed all the basic things related to the bacteria. Since we started the 5 kingdom classification, the very first class kingdom comes into the limelight is the kingdom Monera. But we have discussed about the kingdom Monera, their shell shapes and sizes, their cellular structure, the organization, the kind of ribosomes, the kind of an organelles they possess, and yet you know what this 6 kingdom classification suggests, that the Monerans have not included, or the Carl Voss, what he did, he actually excluded the eubacteria and archaebacteria from the Monera. Why? We are going to discuss that in tomorrow's class, so do not miss tomorrow's class. Please register yourself for the live class. We are going to discuss why archaebacteria and eubacteria are different from the Monerans and why they were given a separately they have been classified, not given a separate kingdom, but they are not kept in kingdom Monera. They are just having a horizontal own division. Okay. So today we have discussed about the bacteria is completely cosmopolitan and they are distributed all over the places because they are completely microscopic. They can be autotrophic, they can be heterotrophic, and kingdom Monera is further divided into 2 sub-kingdoms, which is the archaebacteria and eubacteria, which we are going to discuss tomorrow, that why these 2 sub-kingdoms were important, why the archaebacteria and eubacteria were kept in the separate sub-kingdoms, and bacteria is reproduced mainly by the fission, and they also show the parasexuality. Okay. So children, before we go, let me introduce you to our, this is our Embibe app. So whenever you wanted to have all the solution, all the problems having this one solution, which is the Embibe app, you can learn over here, you can practice over here, you can basically attempt test, and you can always judge your how the basically you have progress eventually learning through the topics, different chapters, different subjects, all at one place. So this is your Embibe app, children, and as soon as you go to the home page, you will see there will be the Embibe live classes, and all the upcoming classes would be basically over here. And if you see this, this is tomorrow's classes. You can register yourself for tomorrow class, and day after tomorrow we'll discuss about the protesta. These are all the recorded session present in the Embibe app. You can learn through different books, different videos, different different questions are already present. Okay, you can go through any subject you wanted. This is me already taking your class. These are the trending videos, these are the videos with solution, these are the Embibe explainers, and these are the world's best curated content. You can go through everything according to your need, according to your particular subject. You can practice, you can create your own test, and you can see how much progress you have made. So children, please go and download the Embibe app and see how much you can explore. So many things, so many good things in the 3D are already there. Some Embibe curated videos are also there by the Embibe expert, and the best part is that even if you belong to any state, you can choose your own curriculum by selecting your own state board. Okay, I will tell you how you can select your own state board. Going to the home page, if you see on your left there is 11 CBSE written. I can select any board, be it Karnataka board, be it Punjab board, Delhi board, Goa board, whichever state you belong to, you can basically select. And as I select I am selecting the Rajasthan board right now. As I press next, I can select my exam, I can select my class which I belong to. So you can also study according to your board syllabus. Okay, all the things were according to your board examination. Okay, moving forward, there's an amazing app which is the Embibe Lens app. This is nothing but an completely visual learning for you children. You just have to scan the text. It will take up all the keywords and will show you all the related 3D objects. Okay, so please go download it and just see everything in 3D. Any textbook, biology, physics, it will show you things in the 3D form. Okay children, we'll meet again tomorrow. With this, at the same time, we'll discuss about archaebacteria and eubacteria, why they're having these sub-kingdoms. Till then, please take care and keep imbibing. Bye-bye.
Keep imbibing. We believe in you.